Good morning to all of you. Right. So, how many of you have searched with respect to tinea, different particular infections? Please let me know. Right. Okay, Raj. Particular. You talk about tinea versicolor. That would be also fine. You unmute yourself and you can just speak what you find it. Right. Or if you want to write, you can write it down. I will speak to all. That will also fine.
Hello, sir.
Yes, Raj. You can speak. Thank you.
Yes, sir. Can I share my screen? મેં લખ્યું છે તો ઈ
ઉભા રહેજો તમને સ્ક્રીન શેર કરવા માટે મારે હોસ્ટ બનાવવો પડશે. Do you want to પછી તમને you can change again, you know how to change to or how to make me host, right?
Yes, sir.
Okay, then I'm making you host. You can share the screen, right?
Okay, sir.
Okay, I'd make you host. You can share your screen.
Just 1 minute.
Yes, fine. I had stopped my screen. You can share.
Yes, sir.
Yeah, thank you.
My screen is visible, sir?
Yes, Raj, it's visible. Great.
Tinea versicolor. Symptoms: patches of skin, lighter or darker than surrounding skin, loss of color, rashes and redness. Diseases are mild itching, liver spots and skin discoloration. Mode of transmission is through skin contact. Detection are microscopy through KOH and culture test. Causative agent is Malassezia furfur. Epidemiology is there is occurs worldwide, but the highest incidence is found in tropical climates, and the most commonly affects young adults and adolescents. Treatment are there are antibiotics, clostrimazole, microzole, and selenium sulfides. Control are good skin hygiene, avoiding excessive heat, sweating, and sun exposure. Thank you, sir.
Great, Raj. Thank you. Thank you for showing interest and finding out some particular symptoms. Right. Okay. So.
તમને host બનાવી દઉં છું.
Yes. Fine. Thank you. Yeah. Okay. So apart from that, there are few responses which have came up. I will read all that responses. Right. Okay. So Priya Solanki has searched Tinea captis. Priya, if you can just elaborate much more on symptoms, I will read it out. Dikshit had searched for Tinea rubrum. Tinea rubrum works very well, fine. Then, okay. Dikshit has searched for Tinea rubrum, and what he has said is disease usually happens in skin and hair. Symptoms are redness and transmission is person to person, right? Narendra has searched for Tinea solium, uh, Tinea solium, and Nehal has found out with respect to Tinea captis. So what she has found is with respect to first symptoms is ringworm and scalp. So ringworm disease of scalp and hair shaft. Signs and symptoms, it becomes itchy, bald patches, common in toddlers and school of children. And how it is spreading, it is spread via contact through an infected hair, like if anybody has and combed that particular hair, and in the comb the spores remains, and the spores actually attaches to the uh, pillows, and that the pillows and even that comb becomes a source of infection, and then it spread further. The treatment, even she has written, Nehal has written the treatment can be ketoconazole. The ketoconazole works very well, and then griseofulvin, uh, even shampoo containing ketoconazole can works very well. So over there it works very well. For Dixit, for Tinea sporum, the treatment can be griseofulvin or itraconazole. Priya has finally found out what can be the symptoms of tinea capticis. It can be scalpy, itchy, scald, bald patches. So if we see with respect to most of the infections are found, almost similar, and they are founded that yes, almost similar, because again it is targeting directly the keratin part only. You see with respect to tinea capticis, tinea rubrum, everything is the signs and symptoms are this itchiness, scalpiness, bald patches on hair and head. So these are all the things. Then what Narendra has found out that there is a Tinea solium species is there. The in Tinea solium, the mode of transmission is it is acquired through humans, and usually the larval stage of the tinea. He has found very nice that larval stage of such parasite is causative agent, and it is found usually in undercooked meat or infected pork. The symptoms, which is totally different compared to any other species, it includes the symptoms like digestive problems, including abdominal pain where the appetite loss has been found, even weight loss and stomach upset has been found. And what can be the probable mode of detection? Either stool examination or CBC count can be done. Epidemiology, overall, globally, 40 to 60 million people are affected. In US, 25% of cattle has been found infected, and but because it is only 25%, it was considered as a low. Thank you, Narendra, for like extensive search with respect to all the species. Tenia solium. Then Priya has said that usually the treatment is again griseofulvin, itraconazole, and terbinafine. Prevention can be properly cooked meat, right, Narendra? Great, great. So I'm really thankful to Raj, Priya, Narendra, then Dikshit, Nehal to at least find out and showing some interest in the particular subject, right? Thank you all for finding and sparing some time. So I think I will again share my screen, and then we'll start our normal session. So yesterday, where we have stopped actually. So we have stopped over here with respect to what can be the probable role of infection still, right? And in that, the role of infection was with respect to, we are talking about the nail bed. Now, in nail bed, what was the problem that beneath the nail, near the nail tip, infection or fungal infection propagates, and it causes something which is an unusual or disorientation of the nails. Usually what the 4 or 5 members have searched, uh, the specific infection was with respect to hairs. If you see the scalp hairs, only with respect to one of the species, it was with respect to digestive system. Rest all was with respect to a specific infection or something associated with an keratin. Similar over here also, the same thing, but totally different aspects we need to understand, right? Further, Tinea captaceus, again, what I think one of the member has searched, but over here, apart from Tinea captaceus, what there are two another species which has been found out. One we call as an microsporum, and second one is an trichophyton. Now, whenever there is a ringworm of any type of scalp, eyebrows or eyelashes, right? Now, please, if you understand it, with respect to scalp, it is common. It can be easily found out. But with respect to eyebrows and eyelashes, this is hardly actually been found out, but it is also very rare. The reason why it is rare and it is rarely found because we usually clean our face 7 or 8 times depending on how much we like our face. But still, if we are not, we are applying different type of creams either to moist our skin or to dry our skin. And if females are usually in that particular eyebrows or eyelashes, they used to have different makeup. Now, even if they see the constituent of that makeup, it prevents the particular bacterial or any type of fungal infection. So, with respect to ringworm, usually it is observed on scalp. Now, whenever there is a combination of these three, like tinea, microsporum, and trichophyton. Now, these three combination makes something and deadly combination, and it directly impacts all these what we call as in scalp, eyebrows or eyelashes. Now, what we called it as this scalp, eyebrows or eyelashes is it enters into an hair follicle part. So fungus grows into hair follicle, and now once it enters or grows into hair follicle, it starts reproducing itself through an hyphal propagation, right? And how does we it looks like? Because an microsporum can give us an fluorescent green color. So whenever you keep such particular hairs which are infected with an microsporum trichophytum, you will always observe that an fluorescent green color has been elicited, right? And once it is elicited, you can easily observe, yes, it has been infected. However, please note down over here, you don't require any type of fluorescence microscope at all to observe the fluorescence. Just an ordinary lamp in a dark room, and if you observe it, you will see a green color, right? If it would be an infected with trichophyton, you will never be able to see any type of green color. If there is a combination of microsporum, trichophytum, and tinea species, then and then you will be able to see what can be the probable coloration or glow with respect to the infection, right? Now, how to culture it? Usually, once you identified, yes, there is a presence of fluorescence, uh, because it is an sporulating form, definitely conidiospores will be present. So what you need to do is you need to just take the hair. Now what you need to do is just keep that hair in a specific media. Once it will form an conidiospores, it will go into a mycelium stage and it will grow into an hyphal or hyphae. From hyphae, it will go into mycelium. Now, over here, we need to understand that there is a pattern of infection. Pattern of infection becomes like specific pattern of conidiophores and hyphae fragmentation. Hyphae means from spores, there is a small particular hair-like structure got what we call as an hyphae, and once it spreads all over the petri dish, we call it as a mycelium. So depending on how does actually an conidio fragments into an hyphae, how does it fragments into a mycelium, we can understand how much infection or what type of infection is. So usually infection has been categorized over into two parts. One we call as an exothrix infection, what we call as an ectothrix, right? And one we call as an endothrix infection. Now, what is ectothrix? Ectothrix means fragmentation of mycelium into conidia called as an anthroconidia. So usually it has been found near the hair shaft or beneath the cuticle where the cuticle has been disturbed, right? So for us, the most important thing to understand it is just a fragmentation of mycelium into conidia, right? So whenever the mycelium is been fragmented, it is usually been further been separated into an conidia. Whereas in endothrix infection, what we call as an endo over here, ecto. So outside, endothrix means something which is inside. So inside would be spores. So that's why the most important thing is called as an arthroconidia. So arthroconidia formation occurs once the hyphae is fragmented, once the hyphae has been disturbed, and then using the hair shaft, it disturbs the cuticle. So over here, mycelia is fragmented to conidia. Over here, conidia leads to an fragmentation of hyphae. That's the only difference between what we call as an exo or ectothric infection, and second one what we call as an endothrix infection. Just an role is opposite. Over there, fragmentation like mycelia is fragmented into conidia, and in endothrix, actually the conidia leads to fragmentation of hyphae. That's the difference, and that's why totally different species came up. Like majority in ectothrix infection, you will see microsporum species only, Microsporum odioni, Microsporum canis, Microsporum fagunum, Trichophytum verrucosum, and Trichophytum magni. Whereas in majority of endothrix infection, you will see it is majorly Trichophyton species, right? So Trichophytum violicum, Trichophytum rubrum, Trichophytum gorvili, all are so just basically on the characterization or fragmentation pattern, we are able to understand is it as an ecto or endo. And then if it would be an ecto, the major dominant species would be Microsporum. If it would be an endo, major dominant species would be Tricho, right? So this is how we differentiate further. Due to the whenever there is a combination of Tinea, Microsporum, and Trichophytum, there is a specific patch or patch of ringworm. It is called what we call as an gray patch ringworm disease. It is an ectothrix common disease, usually found in children between the age groups of 4 to 9 years, but it does not give any type of an inflammatory pattern of symptoms. Usually ringworms, what will happen? There is a specific inflammation. There is a round circle, and near the round circle, the wrinkles are there. And due to wrinkles, actually, you will be able to identify, yes, it is an particularly fungal infection. But over here, there is no inflammation. Now, due to no inflammation, we are not able to bifurcate is it is a fungal or it is something which is associated with a bacterial infection. So usually, but good thing it is usually observed in children. Then there is, because we know there are different type of dermatophytes, this also includes a specific zoophilic and geophilic dermatophytes, which is causing and gray patch ringworms also, right? So what can be the particular treatment of it? Now, uh, the persons the who has contributed, who has just enlisted probable treatment was they found out and griseofulvin was there. Yes, griseofulvin can work very well over here, right? But it usually topical treatments are ineffective. So good thing about the advantage of ectotrix is it can resolve by their own, right? What we call as in gray patch ringworm, because it is happening in the children, it can resolve by their own. Whereas endothrix infections, it can become chronic and may continue into adulthood. So over there, we require a specific either an antibiotic or any type of dermal cream, and that's why over here the most commonly used dermal or fungistatic agent is miconazole and clotrimazole, right? Now it is always been prescribed in a combination of griseofulvin, even though just griseofulvin will not work as efficiently for this type of treatment. But whenever there is a combination of griseofulvin, miconazole, and clotrimazole, it works very well. So that's why over here, whenever we are talking with respect to any type of endothrix infection, usually fungi static agents are prescribed over here, right? Further, if we don't want that to continuously expose the patient with any type of dermal creams, usually scrubs are helpful to clean the scalp, right? And that's why the scrubs which usually screen the dermatophytes or which can prevent the dermatophyte infection are used. So that's why this is usually used. But however, it is not been prescribed where the patients are being treated with porphyria. What is porphyria means? And specific blood pigment is accumulated and in bloodstream and urine, and due to this accumulation of bloodstream and urine, it leads to an hypersensitive reaction, and due to this hypersensitive reaction, it becomes a very chronic sometimes. So that's why usually it has been known prescribed to use scrubs for in patients who are suffering from an porphyria or any type of some particular disorders associated with hypersensitive reactions. This is what normally an Tinea this species infection start with, and what we call as an gray patch is something like this. Now please mark that this is curable disease. It is not like that it is incurable. It is curable. Just and proper medication or dermal cream associated with it should be applied. So definitely it works very well and it is curable, right? Further, if you see it, then there are small patches usually observed. So what it indicates that it indicates that it always be there going to spread in a patches form only. It will never spread as such in a particular specific small circular form, like a small circle, and then there will be wrinkle on the edges, there will be an spores on the edges. It will never actually happen like that. What will happen actually? uh, there will be a small circle, and this patch ultimately will form another patch. This patch again will form to another patch, but it will be restricted to a certain extent of your scalp only. So this is good thing. The advantage is it does not spread all over your scalp. It remains, it restricts itself to a certain portion of the scalp. Now, once that certain portion of the scalp is remain as such, then there is a no particular way that it can spread, right? And that's why this what we call is it is a treatable disease. If it would be have just passed through all over the scalp, it becomes an uncurable, and this how we think that it should be stopped at certain point of time. Then how do we identify ectothrix and endothrix infection, right? And for that, simple, you see this hair, you can see this is where actually due to specific nature of microsporum species, it glows. This is a normal, whereas there is a no microsporum species is there, and these are this particular part of hair where actually the microsporum species is usually observed. So this is the portion what we are talking right now, and this is a normal portion. Further, if you see in depth or zoom it out, what is reasoning for such type of particular glowing is actually whenever it passes, this is what our tissues are looks like. Whenever these tissues are there, you can see these are the patches, right, which is actually glowing whenever you are passing through it in specific length. Now what these are, whenever again you zoom it out, these are bundle of spores accumulated in this particular portion. This is a zoom part of this what I'm marking. So these are actually a bundle of different quantity of the spores. Now whenever there is a high quantity of spores, it will liberate the fluorescence. Now due to this liberation of this particular energy, it will glow itself, and that's how we can identify, yes, it is something which is an ectothrix, and we can differentiate the disease or infection that it is ectothrix or endothrix, right? So this is how we usually differentiate it. Then because we are associated with a specific type of dermatophytes, microsporum or trichophytum species, we need to understand how does it looks actually under microscopy, because this is not like a normal fungus like aspergillus or trichoderma, which has a specific pigment associated with it, right? Usually aspergillus species neither has a specific black pigment associated with it. Aspergillus, uh, other species of aspergillus like aspergillus niger, aspergillus tereus, all have different pigmentation. And how do we differentiate? Because basically on the color, but as soon as we see microscopically, it gives a specificity how does an hyphae and how does an mycelium looks like. So this is how the culture is grown. You can see it is in colorless, it is in white color. So mostly we are not able to judge which type of particularly fungus is there. But yes, as soon as we go for its initial microscopy, we are able to find it out. Now this particular structure looks like this, right? There is a clear hyphae, and this is what we are interested in, how the hyphae is connected with the sporulating structure. And the nice thing about it, if you are understanding it, you will understand the lobe structure which is present inside the spores, right? Now because we talked it is a microsporum or trichophyton, the most important thing is to understand how much lobes are present within that particular sporangium. Now, what is sporangium over here is something which is this structure we called it as, right? And this is one spore. Now, all the spores are further categorized into different lobes. Lobes means you can count how much divisions are there within the spores. How much divisions are there? You can easily find and you can easily calculate it. So over here, if you see, just see 1, 2, 3. You will find similarly in all the spores. You can see 1, 2, 3. Still over here, the differentiation is still going on and still. So how does the first spore looks like? This is how the first initial budding stage of that particular spore looks like. The second budding stage would be similar to like this, right, where it has been just bifurcated into two. The third stage looks like this. What you are able to see in the marked region over here, where it has been filled up, and once it has been filled up, it is getting elongated over here, right? And then once basically on the elongation part, how does it separate itself? You can see as soon as it becomes mature, once it becomes mature, it will separate into like this 1, 2, 3 part, right? So every particular spore further bifurcates it into 3 parts. Further, this is what we call as in microsporum looks like. If we see with respect to trichophytum, because these two species are the most important, microsporum and trichophyton, right? The trichophytum looks like this, similar to a structure of leaf, very similar to a structure of leaf. So this is usually a structure of any type of leaf, and usually leaf is like something like this, right? It is been marked like this. Over here, within the leaves, just like a leaf, it has been lobed into many sublobes. Sublobes means is 1, 2, 3, 4, 5, 6, 7, 8. So usually 7 to 8 lobes can be easily found out, and exactly it is similar to a curved shape leaf. Over here there is no curve over there, it is just like an oblong shape which is there. And over here, instead of oblong shape, there is a specific leaf-like structure which is present over here. This is what we call as a difference between an trichophytum and microsporum species. That it is almost very different when we observe it, right? The only differentiation stage would be initially if we will able to count the number of lobes of number of species associated with, then and then we will able to find out. Further, there is no particular pigmentation associated with the hyphal or the spores. It looks really white only. So there is no particular differentiation with respect to any type of sporulating structure. Only through microscopy only we are able to find out a difference between them and that particular species only. And still, if you want to better visualize it, right, it looks like a connecting bridge, and you can visualize like this. The only thing you need to zoom it out, and once you zoom it out, you will be able to understand overall the structural pattern of the dermatophytes, right? So this is what we call as an conidia or anthroponidia formation on a specific hairs, right? So this is what the conidia is formed, and now the spores are releasing from here. Now because spores are releasing, it usually spreads out, right? So how it spread out, you can see this particular portion is having small spores. So this is the anthoconidia which has been bursted out. This anthoconidia is still in an intact form, and sooner or later it will burst and it will spread the spores. into an hair, right? So further, if we talked about the microsporum species, now microsporum species, in the microsporum genus specific species, what we call as an Auduini, it does not possess any type of telomorph state. Only sexual reproduction has been known. No asexual reproduction has been known over here. It does not possess any type of conidia at all. Usually there is an conidia spores are there, and then it transmit, but usually or till date it does not possess any type of conidia, right? Further, if we want to still characterize it, and if you want to observe microscopically, it has a septate hyphae. It has many septas, or what we call as in lobes, and usually at the end it is pointed, right? It is irregular or non-uniform in the shape. Usually colonies looks very flat, but appearance is very silky. On culture media it is very tight because it is an something an mycelial structure associated with it. And if there is at all any pigment at the maturation stage, at mature stage it becomes an reddish brown in color, and it gives a specific pigmentation. This is how actually it is looks like. So usually whitish in color, but at mature station stages you will always observe such type of and pigments or reddish brown pigment associated with it. So usually it has been found that, right? And if anybody is much more interested in finding different world of fungi or different diversity of fungi and correlation of medically important fungus, right? What will be the probable medically important fungus would be, please visit www.drfungus.org, and in that you find different species of fungi which are very relevant to medical field, and nowadays which has been much more researched also, which is a causative agent of many disease. And you will find with respect to its hyphae, you will find how does it grows, everything you will find it out over there, right? Yes, Ishika. So www.drfungus.org. This uh, this is, or else you can just take the snap of, if you are like take the snap of the particular current slide, it shows which particular website is there, right? Okay. So further, with the same genus microsporum, there is another species what we call as an microsporum canis. Now microsporum canis is different compared to what we call as an microsporum auduini, right? How does it works? Because you can see over here, normally microsporium auduini never reproduce using an asexual. It has been observed only sexual reproduction, whereas in microsporum canis telomorph is present, and due to telomorph it usually works with an asexual. But it has been also found that few of the species which has been mutant species were able to go through an sexual reproduction also. So mating type, if it is minus, then telomorph is present, and if telomorph is present, suddenly it becomes an ascomycetes group, and the name it is called as an arthroderma otei. Arthroderma otei. So it is just that instead of an normal sexual reproduction stage, it goes into asexual, and then it reproduce itself and then propagate and infect any of the human beings, right? So if we see microscopically, Microscopically, this microconidia, which was again absent in Microsporum odeoni, over here it is present. It is very, very abundant. Much more is there. Thick walled, very, very thick, and usually it is curved at the ends, right? And it is sometimes the microconidia are small and clavate, club shaped. This is how it looks like, right? You can see very, very clear Microsporum canis. And if you see the structure, this is how actually and white color clear structure is there. Very structure similar to a normal particular leaf, and even particularly curved at the end. So you can see it is curved at the end also, right? Further, the third species of the same genus, Microsporum gypsum. Now, Microsporum gypsum is something which is having and similar characteristics of what we called an Microsporum odeoni and Microsporium capsis. Now, over here, the similar to capsis, tenomorphs are Arthrodenia gypsum and Arthroderma incurvatum. Now, incurvatum means over here you will see or you will observe the curves or the twining which was there in capsis is absent. So, incurvatum. If it would be curves, then it would be curvatum, incurvatum. Now, what could be the probable microscopical observation? Macroconidia, pigmentation brownish to yellow, and usually the if we observe it on petri dish, it observed like a particular powder, brownish yellow powder. If we sprinkle it, it observes like this, right? So usually it is having 4 to 6 septa cells or cross walls, and it is 40 micrometer, near about 40 micrometer long has been observed. Now, if we stain it further, whenever we stain or go for staining of it, the lactophenol cotton blue or lactophenol, usually micro due to lactophenol or phenol concentration in the lactophenol cotton blue, the microconidia gets collapsed, and due to that we will not able to see the microconidia. And usually the microconidia of this species do not been or are not observed on any type of infected hairs, right? It is usually observed, but it is not usually observed in any type of something what we call as an infected hairs. So unusual, but still it is infective. This is how actually it looks like. You can see it is exactly and powdery brownish yellow, what it has been written over here, that brownish yellow powdery appearance. So brownish yellow powdery appearance, well defined septa. Incurvatum, there is no curves associated, just an normal oblong structure. Whereas if you compare with respect to capsis or canis, you can see it is curved, right? Over here, there is no curve. Overall, it is just a normal particular oblong structure which has been observed over here. And due to that, what will happen? It will never form any type of conidia. When and due to phenol, we will not able to observe many structures perfectly under microscopy. You will see there are many structures which has been deformed. Only few structures has remained. Rest structures has been deformed. This is the hyphae, but still there is a deformation of hyphae. Okay. So with that, we would conclude today's session, because from tomorrow we will talk about the Epidomophyton, another species of dermatophytes, and then we will talk about much more of the Trichophyton also, which is remaining, right? So again, an humble request, if anybody is still interested, they can search for microphyton species or microsporum species. Different microsporum species you can identify, and what are the different cultural characteristics, what are the infections it leads, and once it leads to an infection, you can work you can know different aspects of it, right? So next session, please, those who are interested, please compile for different microsporum species. And similarly, it would be great just if anybody can do like Raj, they can write on paper and they can share the screen. It would be helpful to others also to remember, right? Okay, I am stopping the screen sharing so that I can take the attendance. Total 61 participants are there. So I will speak up the name, those who are present in a second, and then so as far as list of participants are there, Anusha Chawda, Daksh Suthar, Darshak Ramani, great, he is also present. Devi, Devanshi, Dhruvi Chandarani, Dikshit, Diya, who is DK? I am not able to get.
Sir, Darshan bolo.
Okay, Darshan is there, right? Then Dikshit, Diya Rawal, Fadili, German, Fahad, Hanson, Hasti, Hetvi, Himanshu, Ishika, Kennedy, Khushi Parmar, Krishna Maradia, Krishna Vania, Mandeep Singh, Mansi Kalaria, Mansi Mali, Mar, Masti Satani, Mutebi, Nancy, Nandini, Narendra, Nehal, Nelson, Nidhi Gondalia, Nitkita Bada, Paras, Priyanka Dave, Priyanka Raj Detroja, Riddhin Modi, Ritika Unhad, Riya Chandra, Roshni Kerai, Sanjay, Shivam, Shraddha, Shruti Tanya, Tito Umil, Vaishnavi Patel, Vijay, Vivek Jotanya, Wilson, Esha and Zara. Right? I've taken the snapshot of all those who are present, and please, it would be great if you can just come up with the symptoms of microsporum species. It would be really helpful, right? Thank you all. Thank you for attending the session. See you all soon.